What if I told you there's a single ingredient, one you can pick up at any grocery store for less than $1 a day, that when added to your morning coffee, does three things simultaneously that most people over 60 have been told are medically impossible? It reactivates the stem cells your body gradually stopped producing after the age of 50. It cuts off the primary fuel source that cancer cells depend on to grow and multiply. And it burns visceral fat, the dangerous inflammatory fat wrapped around your liver, pancreas, and heart, faster than almost any dietary intervention ever studied in randomized controlled trials. And what if I told you that the reason your doctor has never mentioned this to you has nothing to do with conspiracy, nothing to do with pharmaceutical suppression, and everything to do with the fact that this research has been sitting in peer-reviewed journals for nearly a decade while medical education moves at the pace of a glacier? Most physicians are still practicing medicine they learned in the 1990s. The research I'm going to share with you today is from 2018, 2019, 2021, 2023. It's cutting edge, and it's only now beginning to filter into clinical practice. Hi, I'm Dr. Ryan Mitchell. I'm a physician who has spent over 30 years investigating the intersection of metabolic medicine, cellular regeneration, and cancer prevention, specifically in patients over 50. What I discovered when I started examining how certain medium-chain fatty acids interact with aging biology completely transformed how I think about what my patients consume in the first hour of their day. And today I'm going to show you exactly what the research says, how this works at the molecular level, and the precise protocol used in clinical studies, so you can start tomorrow morning. In this video, we're going to cover 4 essential areas. First, I'm going to explain why the biology of people over 60 makes them uniquely vulnerable to 3 interconnected threats: stem cell exhaustion, cancer cell proliferation, and metabolic collapse. And critically, why these 3 processes are far more connected than anyone has explained to you before. Second, I'll walk you through the 5 biological mechanisms by which one specific type of fat, added to your morning coffee, addresses all 3 of these threats simultaneously, not sequentially, simultaneously. Third, I'm going to show you the clinical studies that prove this works, with exact numbers, sample sizes, institutional affiliations, published journals, because I don't expect you to trust me. I expect you to trust the data. Fourth, and this is the part most people get wrong, I'm going to give you the precise protocol, the exact type of this ingredient to use, the exact amount, the timing, the preparation method, and the 5 critical mistakes that will eliminate most of the benefit, even if you're doing everything else correctly. Quick note before we dive deeper. I receive messages every single week from viewers who tell me they've watched 4 or 5 of my videos and didn't realize they weren't subscribed. If that's you, would you take 2 seconds right now and check? It costs you nothing, and it helps this information reach more people who genuinely need it. Now let's begin with what's actually happening inside your body after the age of 60. Because understanding this is the foundation that everything else builds upon. After the age of 60, 3 distinct biological processes begin accelerating simultaneously. Most people have heard of one of them. Almost nobody understands all 3, and virtually no one, including most physicians, understands that these 3 processes feed each other in a vicious cycle that makes each one dramatically worse than it would be in isolation. Let me explain. The first process is something called mitochondrial senescence. Your mitochondria are the microscopic power plants located inside virtually every cell in your body. They take the food you eat and the oxygen you breathe and convert them into cellular energy, a molecule called ATP that powers everything your body does. When you're 25 years old, a single muscle cell might contain 2,500 fully functional mitochondria, all running at peak efficiency, all generating abundant energy. By the time you're 65, studies using direct muscle biopsies and electron microscopy, published in journals including the Journal of Physiology and Aging Cell, show that mitochondrial density declines by 40 to 50%, and the mitochondria that remain are often damaged, inefficient, and producing excess reactive oxygen species that damage surrounding cellular structures. Your cells are literally running on half power, and they're running dirty. This is why everything feels harder after 60. This is why you experience fatigue after activities that felt effortless at 45. This is why your recovery from exercise, from illness, from stress takes twice as long as it used to. It's not psychological. It's not just getting older. It's a measurable, quantifiable reduction in your cellular energy production capacity. The second process is stem cell depletion, and this is the one that almost nobody talks about, but it may be the most important of the three. Stem cells are your body's master repair crew. When tissue is damaged, when a muscle fiber tears during physical activity, when an arterial wall becomes inflamed, when a section of intestinal lining wears down, when skin is wounded, stem cells are the cells that migrate to the damaged site and regenerate healthy new tissue. At 30 years old, your body maintains robust, active populations of mesenchymal stem cells, hematopoietic stem cells, and tissue-specific progenitor cells that keep your organs in a constant state of renewal and repair. By 65, multiple studies, including research published in Nature Medicine and Cell Stem Cell, measuring circulating stem cell populations in human blood samples, show declines of 60 to 80% compared to younger adults. Let that sink in. The repair crew that maintained your tissues for decades, that healed your wounds, repaired your arteries, regenerated your gut lining, replaced damaged cells throughout your body, is now operating at 20 to 40% capacity. This is why wounds heal slower after 60. This is why arterial walls repair less effectively, allowing plaque accumulation. This is why gut lining regenerates with gaps that allow inflammatory particles to enter your bloodstream. This is why everything breaks down faster than it rebuilds. And here's the part that matters most for what we're discussing today. When stem cell depletion coincides with accumulated DNA damage in a tissue, which is inevitable after decades of oxidative stress, environmental exposure, and normal cellular wear, there are fewer healthy stem cells available to replace damaged cells before those cells can accumulate the mutations that transform them into cancer. Your repair crew can't keep up with the damage, and damaged cells that aren't replaced become the seeds of malignancy. The third process is metabolic dysregulation. After 50 and accelerating after 60, your cells become progressively less sensitive to insulin. The enzymes that break down stored fat become less active. Your body's ability to use fat as a clean, efficient fuel source diminishes, and instead, glucose, blood sugar, becomes the predominant fuel source for almost every cell in your body. This shift toward glucose dependency matters enormously for cancer risk, and I'm going to explain exactly why in detail shortly. But the short version is this. Cancer cells have a fundamental, well-documented metabolic addiction to glucose. They consume it at rates 10 to 50 times higher than normal cells. When your entire metabolism shifts toward a glucose dependency, when glucose is abundant and constantly available, you are creating exactly the metabolic environment that cancer cells need to thrive, multiply, and metastasize. Now here's what nobody explains. These three processes don't just happen in parallel. They amplify each other in a self-reinforcing downward spiral. Mitochondrial dysfunction increases the production of reactive oxygen species, free radicals, which damage DNA. That damaged DNA accumulates in stem cells, reducing stem cell viability and function. Fewer functional stem cells means fewer healthy cells available to replace those with damaged mitochondria, which means the proportion of cells with dysfunctional mitochondria increases, which produces more reactive oxygen species, which damages more DNA. Meanwhile, dysfunctional mitochondria can't efficiently burn fat for fuel, so your metabolism shifts further toward glucose dependency, which feeds any cancer cells that are developing, which depletes metabolic resources, which stresses mitochondria further. It's a triple feedback loop, a biological death spiral, and it accelerates with each passing decade after 50. The critical question is this. Is there a single intervention that interrupts all three of these processes simultaneously? that targets mitochondrial function and stem cell viability and the metabolic environment that cancer cells depend on at the same time with a single daily action? The answer, based on peer-reviewed research published in journals including PLOS ONE, Nutrition and Metabolism, the Journal of the Academy of Nutrition and Dietetics, Nature, and Cell Metabolism, is yes, and the intervention involves a specific type of fat, taken in a specific way, combined with something you are almost certainly drinking every morning already. Now here's where the science gets genuinely fascinating, and I want to explain exactly how this works at the molecular level, because understanding the mechanisms is what will help you commit to this protocol for the 8 to 12 weeks necessary to see meaningful results, rather than giving up in week 2 when the scale hasn't moved yet. The ingredient is MCT oil. Specifically, a purified fraction of MCT oil called caprylic acid, or C8. Let me explain what that means, and then walk you through the 5 distinct biological mechanisms that make it work. MCT stands for medium chain triglyceride. All dietary fats are composed of chains of carbon atoms attached to a glycerol backbone. Long chain fats, the kind found in olive oil, butter, meat, nuts, most dietary fat sources, have between 14 and 24 carbon atoms in each chain. These long chain fats require a complex multi-step digestive process. They're packaged into particles called kilomicrons, transported through your lymphatic system, and eventually, hours later, reach your bloodstream. Along the way, they can easily be diverted into adipose tissue for storage, rather than burned for immediate energy. MCT oil is fundamentally different. The carbon chains are shorter, between 6 and 12 carbons. This seemingly small structural difference changes everything about how your body processes them. MCT oil is absorbed directly through the hepatic portal vein into your liver, completely bypassing the lymphatic system. And in the liver, it's immediately, within minutes, converted into molecules called ketones, specifically a ketone body called beta-hydroxybutyrate, or BHB. Now, most people think of ketones as something that only occurs during extended fasting or strict ketogenic dieting. What most people don't realize is that MCT oil produces significant ketone elevation even when you haven't been fasting, even when you're not following a low carbohydrate diet, even when you ate pasta for dinner the night before. This is why MCT oil is sometimes called an exogenous ketone source. It triggers ketone production regardless of your dietary carbohydrate intake. These ketones, specifically BHB, are the key to everything. Understanding what BHB does in your body explains all 5 mechanisms I'm about to describe. The first mechanism is direct fat oxidation, specifically targeting visceral fat, and it works faster than almost any other intervention studied in clinical research. Researchers at Columbia University, in a study of 49 overweight adults over 16 weeks published in the Journal of the Academy of Nutrition and Dietetics, compared participants consuming 18 to 24 grams of MCT oil daily to a carefully matched control group consuming identical calories from olive oil. Same calories, same protein, same exercise protocols. The only difference was the type of fat. The MCT group lost 1.67 kilograms more body weight than the olive oil group. But what made this study remarkable wasn't the total weight loss, it was the type of fat they lost. Using DEXA scanning and CT imaging, the researchers found that the MCT group showed a 2.9% greater reduction, specifically in visceral abdominal fat, not subcutaneous fat, the relatively harmless fat beneath your skin. Visceral fat, the metabolically dangerous fat wrapped around your liver, pancreas, and intestines. The fat that drives chronic inflammation, the fat that causes insulin resistance, the fat that correlates most strongly with cancer risk, heart disease risk, and early mortality. When researchers used indirect calorimetry to measure what was actually happening in participants' cells, they found the MCT group was oxidizing fat, burning it for energy, at a rate 29% higher than the olive oil group. Their cellular metabolism had shifted into fat-burning mode in a measurable, quantifiable way. Why does this happen? Because BHB, the the ketone produced from MCT oil activates a class of nuclear receptors called PPR alpha. When PPR alpha is activated, it upregulates the expression of genes involved in beta oxidation, the process of breaking down stored fat for energy. Your cells receive a molecular signal that says switch to fat burning mode, and they comply. But this is only the first mechanism, and honestly, it's not even the most important one. The second mechanism is what I consider the most remarkable. It involves mitochondrial biogenesis, the creation of new mitochondria. Remember, between the ages of 20 and 70, you lose 40 to 50% of your mitochondrial density. For years, the medical consensus was that this was an inevitable, irreversible consequence of aging. You could slow it modestly through aerobic exercise, but you couldn't reverse it. The mitochondria were gone. That consensus is now being directly challenged by the evidence. In a study published in PLOS One, researchers in measuring the effects of MCT supplementation on cellular energy production found that MCT oil activates a protein called PGC1 alpha. PGC1 alpha is called the master regulator of mitochondrial biogenesis. When PGC1 alpha is expressed, your cells receive a direct molecular signal to construct new mitochondria. After just 4 weeks of MCT supplementation, participants in this study showed 37% higher expression of PGC1 alpha in muscle tissue compared to baseline. Direct muscle biopsies revealed 28% more mitochondria in those same muscle cells, and their measured exercise capacity, their ability to generate energy under physical demand, improved by 25%. Think of it this way. Imagine each of your cells is a factory that generates electricity. When you were 25, that factory had 100 generators running at full capacity. By 65, you're down to 50 generators, and half of them are damaged and inefficient. MCT oil is like a capital investment that factory desperately needs. It triggers the construction of 30 new generators, and it initiates repair protocols on the damaged ones. Within 8 to 12 weeks, your cellular factories are producing significantly more energy from the same raw materials. That's not motivation, that's not placebo. That's mitochondrial biogenesis, measured directly in human tissue samples. The third mechanism involves stem cell protection and reactivation, and the research here is genuinely exciting. BHB, the ketone produced from MCT oil, has been shown to directly influence stem cell behavior through a molecular pathway that researchers at Harvard Medical School and the Salk Institute have been investigating for nearly a decade. A landmark study published in Nature by Chung and colleagues demonstrated that BHB is a critical regulator of hematopoietic stem cell quiescence. Quiescence is a protective resting state. When stem cells are maintained in quiescence, they're protected from oxidative damage. They conserve their regenerative capacity, and they remain ready to be activated when your body needs them. When BHB levels are chronically low, as happens in people over 60 who are running primarily on glucose metabolism, stem cells exit quiescence prematurely. They become exhausted. They accumulate damage. They lose their regenerative capacity far faster than they should. When BHB levels are elevated, as happens when you consume C8-MCT oil in your morning coffee, stem cells are maintained in this protected quiescent state. They live longer. They retain their regenerative capacity. And when they're called upon to repair tissue, they function more effectively. The molecular pathway is well established. BHB inhibits an enzyme called HDAC3, which in turn activates the gene expression profile that maintains stem cell integrity and longevity. This isn't theoretical speculation. This is measured in human biological samples using flow cytometry and single-cell RNA sequencing. The pathway is documented. The mechanism is understood. In practical terms, the simple act of consuming MCT oil in your morning coffee is doing something at the cellular level that almost no pharmaceutical intervention achieves. It's helping preserve and reactivate the repair crew your body depends on to maintain healthy tissue and prevent the accumulation of cellular damage that leads to cancer. The fourth mechanism is where this becomes directly relevant to cancer prevention. And I want to explain the science carefully, because it is specific and it is important. In 1931, the biochemist Otto Warburg received the Nobel Prize in Physiology or Medicine for discovering a metabolic characteristic that he observed in virtually all cancer cells. Warburg found that cancer cells consume glucose at rates 10 to 50 times higher than normal healthy cells, and they ferment that glucose into lactate even when oxygen is abundant. This is called aerobic glycolysis, or the Warburg effect. Normal cells, when oxygen is available, burn glucose efficiently through oxidative phosphorylation in the mitochondria, extracting maximum energy. Cancer cells prefer the faster, far less efficient fermentation pathway, regardless of oxygen availability. For decades, this was treated as a curiosity. An interesting observation without clear therapeutic implications. What researchers have since demonstrated, including the work of Dr. Thomas Seyfried at Boston College and Dr. Dominic D'Agostino at the University of South Florida, is that this metabolic dependency is not incidental. It's central to cancer cell survival, and it creates a specific exploitable vulnerability. Cancer cells, because they have dysfunctional mitochondria, cannot efficiently use fat or ketones for energy. They are metabolically inflexible. They require glucose. When you reduce glucose availability and increase ketone availability, you create a metabolic environment where your healthy cells, which have functional mitochondria, can use ketones efficiently and thrive. Meanwhile, cancer cells are being starved of the glucose they depend on. Healthy cells adapt. Cancer cells are stressed. A study by Poff and colleagues, published in PLOS One, demonstrated that ketogenic intervention combined with ketone supplementation significantly reduced tumor growth rate and extended survival in validated animal models of metastatic cancer. The mechanism was confirmed to be metabolic, reduced blood glucose, elevated ketones, a systemic environment that favored normal cells over malignant ones. Now, I want to be precise here. Precision matters. I am not telling you that MCT oil cures cancer. The human clinical data on MCT oil as a standalone cancer intervention is preliminary. Anyone who tells you otherwise is either misinformed or trying to sell you something. What the science does clearly establish is this. MCT oil produces ketones. Ketones reduce blood glucose. Reducing blood glucose while maintaining cellular energy through an alternative fuel source creates a metabolic environment that is fundamentally hostile to cancer cell metabolism. This is not speculation. This is organic chemistry. This is cell biology. The question is one of degree. How much impact does the level of ketosis produced by daily MCT oil consumption have on cancer risk and cancer progression? That question has been actively studied. What's already established is the direction of effect. And what I can tell you from the research is that people who maintain higher circulating ketone levels show lower levels of the inflammatory markers that drive cancer progression, lower fasting insulin levels that correlate with reduced cancer risk, and better mitochondrial function that supports the DNA repair mechanisms preventing malignant transformation. You're not just removing fuel from cancer cells, you're rebuilding the cellular architecture that prevents cancer from developing in the first place. The fifth mechanism explains why the combination of MCT oil specifically with coffee is more powerful than either compound alone. Caffeine increases fat oxidation by approximately 29% in adults. The mechanism involves inhibition of an enzyme called phosphodiesterase, which elevates cyclic AMP inside fat cells, which activates hormone-sensitive lipase, the enzyme that releases stored fat from adipocytes into the bloodstream. In simple terms, caffeine opens the doors of your fat storage cells and tells them to release their contents into circulation. But here's the problem. If that released fat isn't burned immediately, it simply recirculates and gets repackaged back into fat cells. You need an active metabolic demand, something consuming that mobilized fat to capture the benefit. That's exactly what MCT oil provides. The ketones from MCT oil create immediate fuel demand. When fat is being mobilized by caffeine, your liver and muscles immediately begin oxidizing ketones. The metabolic furnace created by ketone production is waiting for the fat that caffeine releases. Research measuring the combined effect found that caffeine alone increased fat oxidation by 29%. MCT-derived ketone elevation increased it by approximately 25% independently. Combined, the effect was synergistic, 51% increased fat oxidation, more than double what either compound produces alone. The two mechanisms are perfectly complementary. One releases fuel from storage. The other creates demand for that fuel. Together, they create a fat-burning environment that neither produces in isolation. This is why timing matters critically. Consuming your MCT coffee first thing in the morning, in the naturally fasted state you enter during sleep, captures all 5 of these mechanisms at their peak effectiveness. Insulin is at its lowest point. Glucose isn't competing with ketones for cellular uptake. Fat oxidation pathways are primed, and BHB levels, which naturally rise slightly during overnight fasting, are boosted by the MCT oil at exactly the moment when your stem cells, your cancer metabolism, and your fat oxidation systems are most responsive. Now let's talk about exactly how to implement this, because the protocol matters, and there are specific mistakes that will eliminate most of the benefit, even if you understand the science perfectly. Not all MCT oils are equivalent. This is critical. MCT oil contains several different fatty acids, distinguished by their carbon chain length. C6, caproic acid, converts to ketones extremely rapidly, but causes significant digestive distress in most people. Avoid products high in C6. C8 caprylic acid is the gold standard. It converts to ketones faster than any other MCT fraction and is well tolerated by most people. This is what you want. C10, capric acid, converts more slowly than C8 but is effective and well tolerated. A C8-C10 blend is acceptable. C12, lauric acid, is where most people go wrong. Lauric acid is technically an MCT, but it behaves metabolically much more like a long-chain fat. It does not convert efficiently to ketones. It does not provide the same metabolic benefits. Most inexpensive MCT oils are predominantly C12 because lauric acid is the cheapest to produce. These products will not produce meaningful ketone elevation. When you purchase MCT oil, read the label. Look for products that are pure C8 or a blend of C8 and C10. Avoid any product that is primarily coconut oil or that lists lauric acid as the primary component. The higher quality C8 products cost more. The difference in metabolic effect is substantial enough that the investment is worthwhile. Start slowly. This is not optional. Begin with 1 teaspoon, approximately 5 milliliters, about 5 grams, on your first morning. Maintain this dose for 3 to 4 days. If you experience no digestive upset, increase to 1 tablespoon, 15 milliliters, approximately 14 grams. This is the dose used in most clinical studies. Maintain this dose for 1 week. If you're tolerating it well after that week, you may increase to 1.5 tablespoons if desired. The maximum studied dose with consistent benefits is approximately 25 to 30 grams per day. Do not exceed 30 grams daily. Do not start at a high dose. Why the gradual increase? MCT oil is processed entirely in your liver. If you overwhelm hepatic processing capacity on day 1, you will experience loose stools, cramping, or nausea. This does not mean MCT oil doesn't work for you. It means you exceeded your liver's current processing capacity. Most people who claim they can't tolerate MCT oil started at too high a dose. Give your liver time to upregulate the enzymes needed to process medium-chain fats efficiently. This is the mistake I see most frequently, and it eliminates a substantial portion of the benefit. If you simply pour MCT oil into your coffee and stir with a spoon, the oil floats on the surface in a visible layer. When you drink it, you're consuming layers of pure oil separated from the coffee. The absorption is poor, the taste is unpleasant. The metabolic effect is significantly blunted. You must blend it. Use a countertop blender, an immersion blender, or a high-powered milk frother. Blend your coffee with the MCT oil for 15 to 20 seconds until the mixture is completely uniform and creamy, similar to the texture of a latte. This creates a stable emulsion where the oil droplets are broken into microscopic particles coated with coffee's natural surfactant compounds, allowing for uniform absorption throughout your digestive tract. Optional, but recommended, add 1 teaspoon of grass-fed butter or ghee. This enhances the satiety effect substantially, improves the texture and taste, and extends the duration of ketone production. It also adds butyrate, a short-chain fatty acid that supports gut lining integrity and intestinal stem cell health. Consume your MCT coffee within the first hour of waking before eating anything else. Then, and this is important, delay your first solid meal by 2 to 4 hours if possible. Don't force this. Most people find that the combination of caffeine, MCT oil, and butter produces sustained satiety and mental clarity that makes delaying food effortless. You won't feel hungry. You won't feel deprived. During this 2 to 4 hour window, your body is burning stored fat, producing ketones, activating PGC1 alpha for mitochondrial biogenesis, maintaining BHB-mediated stem cell protection, and creating a glucose-depleted environment hostile to cancer cell metabolism. This is your metabolic window. Protect it. Don't break it with food, especially carbohydrates, which will immediately spike insulin and terminate ketone production. For maximum effect, combine your MCT coffee protocol with these practices. First, maintain a modest calorie deficit of 300 to 500 calories per day below your maintenance level. MCT oil substantially increases satiety, making this easier than it sounds. Count the MCT oil's calories, approximately 130 calories per tablespoon, as part of your intake. Do not simply add MCT oil on top of excessive caloric consumption and expect fat loss. Second, incorporate resistance training 2 to 3 times per week. This provides the mechanical signal your body needs to interpret mTOR activation from ketones as muscle building rather than simple tissue maintenance. You don't need a gym. Bodyweight squats, resistance bands, and moderate dumbbells with compound movements are sufficient. Taking your MCT coffee 60 to 90 minutes before exercise provides sustained energy without glycogen dependence. Third, consume 20 to 30 grams of protein within 2 hours after your workout. This provides the raw materials for the muscle protein synthesis that ketone-activated mTOR is signaling for. Let me tell you exactly what to expect and when, because managing expectations correctly determines whether people persist through the protocol long enough to see results. Weeks. Q1 to 2. You'll notice increased morning energy and mental clarity before you notice any change in body composition. Ketones are a preferred fuel for neurons. Many people describe a dramatic reduction in the mid-morning mental fog they had attributed to normal aging. This is real. This is measurable. Your brain is responding to a cleaner, more efficient fuel source. You'll likely notice reduced morning hunger, the ketone-driven satiety effect. Scale weight may drop 1 to 2 pounds, mostly water associated with glycogen reduction. Weeks 3 to 8, fat loss becomes measurable. Expect 1 to 2 pounds of fat per week, totaling 4 to 12 pounds at the 8-week mark. Body composition changes become visible, primarily reduction in abdominal circumference. If you're doing resistance training, strength will hold constant or improve, despite calorie deficit. This is the MCT-mediated muscle preservation effect. Months 2 to 3, mitochondrial biogenesis effects fully manifest. This is when people describe feeling like they have the energy they had 10 years ago. That's not placebo, that's 28% more mitochondria in your muscle cells. Biomarkers improve. Triglycerides typically drop 15 to 30%. HDL rises. Fasting insulin decreases. If you have active liver disease, consult your physician. If you have type 1 diabetes, you need supervision due to ketoacidosis risk. If you have type 2 diabetes on insulin or sulfonylureas, monitor glucose closely. MCT oil improves insulin sensitivity and may require medication adjustment. For most people over 60 without these conditions, MCT oil is extraordinarily safe and evidence supported. Look, aging after 60 is not a sentence. The patients I've watched transform their metabolic health were not genetically exceptional. They made one specific evidence-based change to their daily routine and stayed consistent. Start with 1 teaspoon of C8 MCT oil, blended into your morning coffee tomorrow morning. Give it 8 weeks. If this analysis was valuable, subscribe and like this video. Next week, I'm covering the emerging research on time-restricted eating combined with this MCT protocol, including specific data on fasting and cancer metabolism that almost nobody's discussing openly. Comment below with your biggest question. I read every comment, and your questions shape what I research next. Keep investigating. I'll see you in the next one.